In this video, we're going to explore different models of the membrane. So we're talking about the plasma membrane, cell membrane that surrounds the cell, as well as all the internal membrane structures within the cell. And we're going to think about how scientists have used the best evidence available at the time to create a model, and we can accept or reject that model based on how well it fits the available evidence. And obviously, as technology improves, we might find a new piece of evidence which will then cause us to adapt an existing model. It doesn't mean that the model before is useless. We just build on that model, taking the aspects that work with all available evidence and move forward to give us our best guess at visualizing how something works or how it's structured. So we go back really with membrane models to 1915. And at that time, we were able to take red blood cells. So here's a little image of some red blood cells moving through an artery or vein. And red blood cells do not have very much inside them. They are destined to die. They are full of hemoglobin. And when you put them in water, they burst. So you can take a red blood cell sample and you can burst open the red blood cells. So all you are left with is the membrane. And this is great because it gives you lots of membrane to analyze. And when they analyzed this membrane, they discovered that membranes were made of lipids and proteins. So lipids are fat molecules and proteins are strings of amino acids. So that was where they were at in 1915. The next milestone we'll look at is 1925. And in 1925, two Dutch scientists decided to take a closer look at the lipids. And they discovered that these lipids were actually shaped a little bit like this crazy thing here. This is a phospholipid. And a phospholipid is made up of a phosphate head at the top, that little purple blob, and that makes the head hydrophilic. It really is well attracted to water. And that hydrophilic head, I'll just add a little arrow for you. The hydrophilic portion here interacts with water and attracts water. The other end of the molecule, these two tails, are hydrophobic. That means that they repel water and they will not associate or dissolve in water at all. So the bottom half of this molecule is hydrophobic. And because of this, the phospholipids actually combine to put the hydrophobic tails on the inside and the hydrophobic heads on the outside. So you end up with a structure that looks a little bit like this, with a head and its two tails that associate on the other side with another phospholipid. So you can see that these phospholipids will form a bilayer. So I've got another little image here of a bilayer with lots of phospholipids associating. In the middle, we've got the hydrophobic bit. So all the hydrophobic tails are in the middle here. And all the hydrophilic bits, the heads, are on the outside. So they're on the outside on this side of the layer and on this side of the layer. And that means that we can have water on both sides of this membrane, and it's a very stable structure. So that's a phospholipid bilayer. Next, we move to look at some questions that this model does not address. What is it about these pure phospholipid bilayers that makes it different to real membranes? And the key thing is that they don't stick to watery solutions like a real membrane does. Also, we seem to have ignored the proteins. Where do they come in? So our next date to think about is 1935. So in 1935, two experimenters called Dawson and Danieli, they looked at the problem of the missing proteins and they proposed a sandwich model. Their model looks like this. They have the phospholipid bilayer in the center. So here are my phospholipids in the center, forming a two-layer structure with the hydrophobic bit in the middle and the hydrophilic bit on the outside. And they decided to place the proteins on the outside of this phospholipid layer. So they put the proteins on the outside. They called this the sandwich model. So it was the lipids in the center and the proteins acting as the bread around the outside. This was just a guess. They didn't know. This is before the time of electron microscopes. So in the 1950s, when electron microscopes started to become a thing and they could make some images with it, so that's an image of an early electron microscope there, they were able to actually look at plasma membranes, and they were pleased to see that all the new evidence that came with the use of an electron microscope was consistent with the Davson and Danieli sandwich model. Great. Unfortunately, by the end of the late 1960s, better techniques with the electron microscope meant that we started to realize that membranes are not uniform. So for example, the plasma membrane is about 7 nanometers wide, whereas mitochondrial membranes are only 6 nanometers wide. Plasma membranes do look a bit like the Davson and Danieli model, but mitochondrial membranes do not. They look like sort of beads on a string rather than a 3-layer sandwich. So there are some problems arising here. Let's have a look at what happened next with the development of this model. So we move now to 1972. So not very long after these questions in the late 1960s are being posed. And we have two collaborators, Singer and Nicholson, and they propose a fluid mosaic model. So this time they take the proteins and they embed them across the fluid model, across the lipid bilayer and within the lipid bilayer. So you can see them in this image here. Here are some proteins. So we've got a protein going transmembrane across the membrane from the top to the bottom rather than being on just the outside. And here we've actually got a channel here. Sometimes proteins only go partway through, so this one's only on the inside and partway into the middle. And in addition to that, they've added some carbohydrate chains attached either directly to lipids or onto proteins. So you can have glycolipids and glycoproteins. That's what we call something that's had carbohydrate added to it. This helps with stickiness and helps to address one of those earlier problems of why phospholipid membranes don't stick to water if they're pure. Thankfully for Singer and Nicholson, when even more advanced techniques with electron microscopy came about, the use of something called freeze fracture, which is where you take a membrane and you pull it apart along the middle of the phospholipid bilayer and kind of peel it up and down, you can actually see holes left where proteins pushed through and lumps where proteins have pushed through. So this freeze fracture backed up Singer and Nicholson's model and continues to do so. So this fluid mosaic model is the model we use today. And actually, further evidence that we have now seems to imply it's probably more mosaic than fluid. So it has more proteins than lipids. And that the proteins really do move within the layers, but they tend to associate in clusters. So this is still our model with just a few tweaks. And maybe as we move forward, we'll be able to be even more specific using maybe fluorescence microscopy to tag the movement of these proteins and lipids and actually see how often they move and how quickly they move and how far they move.